Hello everybody, my name is Tricia Levi, and today I will be discussing my research to answer the question, are gravid sandflies afraid of predatory mites? The short answer is they should be. I'm going to introduce a few key points before I look into my methods and results. So the species of sandfly that I used for my research is Phlebotomys papatasi. They are the vector of Leishmaniasis major. The females must blood feed in order to produce eggs. They have a few major life stages here, and there are some common control and prevention methods, such as chemical insecticides and standard repellents. There are a few alternative or green sandfly control approaches that have been used for these thus far. Feeding through systemic control, attractant-based control, or bringing the insect to the insecticide, and a more novel approach, which is the push-pull control method. We are pushing the flies away from potential breeding sites and luring them to lethal traps. So we know what attracts them, however, we don't really know what repels them. So here's where the predatory mites come in. We have identified these mites as Stratiolaelaps scimitus, and we know that they are a potential natural enemy of the sandflies. They are a major pest of the colonies, but they are also used as a biological control pest in agricultural settings, and they can also survive for months if adequate food is present and under extreme conditions. So my study questions include, are Stratiolaelaps scimitus mites effective predators of sandflies, and can gravid female sandflies detect and avoid these mites? So to answer my study questions, I have a couple aims. One is to understand the feeding preferences of the mites, and two is to conduct attraction and oviposition bioassays to determine if the females avoid the sites containing the mites. My hypothesis for aim number one is that mites will prefer to prey on the younger larval stages, and my hypothesis for number two is that females will avoid the sites containing the predatory mites. Moving on to my methods. So for the feeding preferences of the mites, I used these 24-well culture plates. I conducted 3 replicates. Within these wells was 1 sandfly at a various life stage and 1 mite. So each life stage was allowed to have 4 wells, meaning that there was a total of 12 wells among all 3 replicates. Each replicate ran for 48 hours, and I checked and recorded the changes every 24 hours. So here are some actual images that I took through the microscope of each life stage. You can see that there is a gradual growth through the instar stages, the pupa stage, and then the adult stage, where I actually took the wings off the flies to prevent them from flying around the wells. So for aim number 2, I chose to do a 2-choice oviposition or an attraction bioassays. Within these, I exposed the gravid females to a pair of cups, one that had 10 mites, which is the treatment, and one with no mites as a control. And then I allowed the females to make a choice. For the attraction bioassays, they could either be attracted to or repelled from the sites, and for the stimulation bioassays, they could either lay eggs or not on the sites. So for task 1 of aim 2, for the attraction bioassay, I conducted 2 replicates using 12 free flight cages of each replicate. Within each cage was 20 gravid females and these 2 Nalgene pods, each containing a wet sand cup and a microcentrifuge tube, one with mites and one without. On top of the cups were a piece of sticky mesh as a sticky trap. The runtime for this was 24 hours of acclimation period without the cups, and then 6 hours with the cups in the cages. Task 2 was the oviposition stimulation bioassays. So for this, they were very similarly set up. However, one side had the sand cup with a piece of filter paper and the microcentrifuge tube in it, and the other side had crushed mites, meaning that they were crushed directly onto the filter paper rather than inside the tubes as alive mites. Here I had 4 replicates of each one, with 5 pots in each replicate, and they also contained 20 female sandflies with a piece of mesh on top to keep them in, and I ran each of these for 72 hours. Now moving on to my results. So for my mite feeding preferences experiment, I decided to combine all my data for all my replicates and create a percentage bar graph here. So you can see for the percentage eaten, the first and second instars had a very high amount eaten. For the percentage dead, which combined naturally dead and eaten, you can see there is a very good gradient from through all the larval stages, and then adult peaks up at 91%, mostly because they died naturally due to their wings being taken off. So here I have a couple photos I took through the microscope of predatory mites that have actually consumed larvae. So the first image here shows a predatory mite that had partially consumed a first instar larva, and it had eaten most of its body. Also, on the other side, we have the predatory mite and the partially eaten second instar larva. You might notice that the body is not completely consumed because I believe that the mite could not eat the whole thing, so it just ate the inside. So my attraction and repellence experiment results show that there is very little significance in the choice that the flies made if they wanted to land on the treatment or control side. This is consistent among both replicants, however there is a difference between the two. Replicant 1, the flies were 9 days post-blood meal, meaning that most of them actually died in the 24-hour acclimation period, so I adjusted for replicant 2, and many more survived, yet our results are very similar for both. Now we're seeing a very similar pattern for our oviposition and stimulation experiments. Our t-test values are showing very little significance. There's a slightly higher amount of average egg counts for the treatment side for both live and crushed mites. However, this doesn't indicate that they have a preference for this side. So on to my conclusion. So I concluded that predatory mites preferred to consume young sandfly larvae over the older stages of the sandflies. Also that gravid sandfly oviposition response is not affected by the presence of the predatory mites. Why does this matter? Well, there's a couple reasons. In the lab, it matters because we know that it is important to eliminate the mites, especially in the jars containing the early stages. And in real-life application, we can actually use these predatory mites as an effective green control agent by applying them to potential breeding sites such as cow barns and chicken coops. Acknowledgements, thank you very much.